Modern debates often obscure a much older truth. Long before it was a scheduled substance, cannabis sativa served as a foundational technology for human civilization. To trace the actual history of our partnership with this plant, we have to look past current political rhetoric and into the archaeological record. The investigation starts here, in the high-altitude permafrost of the Pamir Mountains, a rugged geographic bottleneck that connected disparate ancient cultures. Tracing the historical use of any flora is difficult because organic plant matter typically degrades in the soil. Finding physical proof of ancient drug consumption requires highly specific environmental preservation. These ancient nomadic tribes buried their dead in the high-altitude permafrost, effectively flash freezing their material possessions for millennia. By combining modern paleogenomics with forensic chemistry, researchers can extract ancient DNA and scrape charred residues from these artifacts to piece together exactly how early humanity utilized the plant. Around 12,000 years ago, as the Pleistocene ice gave way to the Holocene, early agrarian settlements in East Asia first began domesticating wild cannabis. This phylogenetic tree charts the plant's genomic history. It shows how ancient farmers took a single wild basil population and pushed it into two distinct genetic lineages. Looking at the top branch, one group of farmers selectively bred the plant for industrial traits. They targeted specific genes, like KNAT1, which inhibits branching. This forced the plant to grow tall and straight, optimizing it for fiber extraction. On the bottom branch, other communities bred for psychoactive traits. They selected for genes like NDL2 to encourage heavy branching, maximizing the surface area available for the plant's resin-producing flowers. This anatomical diagram breaks down the industrial hemp stock. Ancient communities utilized a process called retting, soaking stalks in water, to dissolve pectins and strip away the outer bark. This yielded the high-tensile bast fibers necessary to create durable textiles, maritime rigging, and early paper. Through intensive artificial selection, early human agriculture engineered a wild weed into separate foundational technologies that enabled early civilizations to expand and communicate. Cannabis did not spread across the globe through natural ecological drift. It traveled in the cargo holds and saddlebags of ancient merchants. This map tracks the early diffusion of the plant. Nomadic horsemen, particularly the Scythians, acted as critical vectors, carrying seeds from the Altai Mountains westward across the Eurasian steppe. As the plant migrated, its application adapted to the specific culture that cultivated it. Medical papyri revealed that ancient Egyptian healers utilized it clinically, applying it as a topical poultice to cool inflammation and treat swollen joints. In Vedic India, the approach was highly spiritual. Hindu priests ground the leaves into an ingestible paste called bhang, consuming it during major religious festivals to achieve a state of meditative purity. The plant proved to be highly versatile, serving as medicine, sacrament, or industrial raw material, depending entirely on the civilization holding the seeds. While industrial and medical uses are heavily documented, proving that ancient people purposefully inhaled psychoactive smoke is a massive forensic challenge. We have to look at highly specific artifacts, like the 2,500-year-old fire-scorched braziers discovered in the Jirzankal Cemetery. Inside a living plant, the chemical compounds exist as non-psychoactive acids. It requires direct heat, a chemical reaction called decarboxylation, to convert those raw acids into the intoxicating molecule, THC. Over thousands of years of burial, that THC undergoes oxidation. It gains extra double bonds and degrades into a stable molecular fingerprint, known as cannabinol, or CBN. This infographic outlines how forensic chemists find that fingerprint. By washing ancient artifacts with chemical solvents and pushing the organic residue through a mass spectrometer, scientists can separate the compounds. For the Jirzankal braziers, the readout produced an undeniable spike for CBN. That distinct chemical signature confirms that mourners over 2 millennia ago were deliberately burning specific high THC strains during mortuary rituals. This graphic details recent breakthroughs that are currently rewriting the plant's evolutionary timeline. In 2025, researchers at the Salk Institute mapped the comprehensive cannabis pangenome, looking deep into the genes responsible for cannabinoid production. They found that millions of years ago, chaotic viral invasions colonized the genome, triggering massive gene duplications. Moving to the middle panel, researchers in 2026 used digital DNA modeling to peer further back into that evolutionary history. Through ancestral sequence reconstruction, scientists successfully resurrected extinct cannabis enzymes in a lab. Unlike modern enzymes that specialize in producing just one compound, these ancient versions were promiscuous multitaskers, churning out multiple different cannabinoids simultaneously. These discoveries demonstrate that the plant's chemistry was born from millions of years of evolutionary chaos, a natural process that ancient humans eventually harnessed and fine-tuned through agriculture. Human intervention has transformed this species into a biological multi-tool, engineering its chemical and structural traits to meet the specific requirements of every civilization it touched. Looking at the final panel, massive new studies confirm that our historical entanglement with the plant goes incredibly deep, identifying specific variants within human DNA that influence our biological response to it. The 12,000-year record of this plant is etched into its own DNA and reflected in our biological reception of its compounds, a partnership that has persisted from the first Neolithic crops to the most advanced molecular sequencing of the modern age.